So good afternoon, everybody. Good morning, everybody. So welcome to the first lecture on Introduction to Computational Neuroscience course. So I know you're all interested in neuroscience, and it's becoming very popular, important field by the year. Its importance is growing by the year. The amount of work, experimental work in neuroscience has been expanding. Lots of new labs are coming into picture. And the amount of data that's being generated is also growing constantly, and funding is also growing. And there was a time when it was in millions of dollars. Now it's billions, and even tens of billions are being poured into neuroscience. And you constantly see neuroscience news items appearing in media. There's a lot of media buzz about neuroscience research and so on. In spite of all the glitz and all the popularity, there are some serious problems in neuroscience, which are sometimes highlighted, but very often people like to just shove it under the carpet. So that's because the way neuroscience as a field of research, as a field of science, as a branch of science has progressed, poses some interesting challenges. Because so I'm sure you're all from some engineering branch, right? Because or maybe you're in biotech, but biotech is also a branch of engineering. It's a biological engineering. Now, if you look at the branch of engineering, there's a typical pattern of progression that you will notice. Let's take some of the more ancient, sciences, like for example, astronomy or building sciences. So there was a time when people used to make observations of the planets and stars and all, and they would fit some empirical formulae. So there was a lot of empirical kind of an approach to the science to handling the data and all that for a long time. Look at astronomy, it's millions of years, sorry, millennia. So similarly, building science also has millennia of history. But both in astronomy and building sciences, like astronomy, they didn't know about Newton's laws and calculus and laws of mechanics, all that until about 200-300 years ago. So similarly, in building sciences, they built pyramids, but they didn't know about laws of structural engineering, laws of mechanics, and how do you calculate, right, the force of gravity and things like that. What is Young's modulus or bulk modulus? All that they didn't know. They just had lots of empirical formula, and they built a lot of buildings. So when we entered the era of modern science, we were able to create a very solid foundation of mathematics, right, to that body of knowledge, or to that understanding, to the understanding of that aspect of nature. And with that mathematical framework, we are able to develop applications, and we are able to use that knowledge for practical applications, for the benefit of society and things like that. So this is a typical progression, this pattern of progression that you see in any of the engineering branches that you're familiar with. But when it comes to neuroscience, something interesting has happened. So I'll talk about history of neuroscience in the next class, in tomorrow's lecture. So there is a lot of progress in science, and all the progress that happened in other fields of science, like electrophysiology and simple cytology, anatomy and so on, has contributed to progress in neuroscience. But what is happening is when it comes to the creation of a theoretical foundation, right, which is a major breakthrough, major stage, a milestone in any branch of science, things are a bit shaky when it comes to neuroscience. So that poses some interesting problems, because this course is about, it's an introduction to computational neuroscience or mathematical neuroscience, because maths and computation go hand in hand. Without a mathematical foundation, you cannot have a computational framework. So when it comes to that kind of a mathematical or theoretical foundation, things are a bit shaky, because unlike, let's say, the laws of thermodynamics, right, if you want to develop some heat machine and all that, right, or a thermal power plant, you need to have laws of thermodynamics. They're fundamental, and there's no uncertainty, there's no doubt about that. Or if you're an electrical engineer, a power systems engineer, you start with the basic laws of electricity and, right, Maxwell equations and or Kirchhoff's laws and all that. And after that, everything follows from that. But if you are a neuroscientist, unfortunately, we still don't have a good grasp of what are those fundamental laws of the brain from which everything can be derived. So therefore, there's a lot of uncertainty, a lot of confusion, and a lot of vagueness, right, in the way we understand systems in neuroscience. Right. So let us talk about some of these fundamental problems, because the objective of the course is to present a theoretical, mathematical, and computational framework to explain brain. What I want to say is such a framework is there, but it's not great. Whatever is available out there right now is not great, right? And there are serious problems. So with this note, I want to develop on this idea, right, in this class. And tomorrow we'll look at history of neuroscience. So like I was saying just a moment ago, the reason probably some of you have taken this course is that all the buzz, all the popularity and glamour that brain enjoys in popular media, because people make these kinds of hyperbolic statements about brain, and that kind of catches attention of a lay reader. So for example, it's very often been said that the number of neurons in the brain are more than the number of stars in the Milky Way galaxy. The thing is, adult brain has about 100 billion neurons, and Milky Way galaxy has about 100 billion stars. Or actually, the better estimates go anywhere between 100 billion to 400 billion stars. Actually, the brain doesn't have 100 billion stars, even adult brain. So there was a paper in Nature a few years ago, which says that one of the greatest myths of the brain is busted. What is that myth? All along, we were so misled in believing that brain has 1 billion 100 billion stars, but actually brain has only 86 billion stars. Sorry, 86 billion neurons. So point, and big deal. I mean, 100 to 86 is not a big difference. It's only like a minor detail. I mean, if you ask me. But point is, these are the kinds of things that people talk, to hype up the whole field. And secondly, here the comparison with Milky Way galaxy number of stars is completely misleading. So what? What if the number of neurons in the brain are comparable to the number of stars in the Milky Way galaxy? Right? I mean, it's like saying the number of I don't know, carbon atoms in my thumb are more than the number of stars in the Milky Way galaxy. So what? I mean, what kind of comparison is it? Or number of sand particles in Marina Beach are more than the number of stars in the Milky Way galaxy. So what? Right. So point is, these kinds of comparisons, they sound very like outstanding and extraordinary, but they don't have much meaning. They don't know what they're even conveying. So similarly, another statement that you often find in popular writings is that the brain is the most complex object in the universe. Now, first of all, how many objects have you seen in the universe? How much of the universe have you seen? Man hasn't gone beyond the moon. And if you just depend on telescopic observations, you can't see kind of objects. I can't see an object on Mars. I mean, I see rock and sand and so on. Right. So, what is this complex object, with which I'm comparing, in the universe, with which I'm comparing the brain? So there are not that many out there, right? And objects on the Earth depends on how you compare. So it's like a small kid saying, I have the greatest mom in the whole world. I mean, how many moms have you seen? So I think the thing is, small children can talk like that because they're forgiven for that. But a professional scientist cannot make such strange comments. But you see this all the time. Now, why is there so much hype in the first place? So let me give some reasons. One reason is more practical and probably even political, because we are in the era of big data, right? And a lot of technology is being developed every day. And the new technology will let you make even more fancy observations. And so if you there's a time when we used to use 3 Tesla magnet for an MRI scan. Now we have 7 Tesla magnet, so you get even higher resolution scans, right? Or a single photon microscope was given way to 2 photon microscope, and later we have 3 photon microscope, and even more detail, and so on and so forth. So everywhere you have these experimental techniques which are becoming finer and finer and more precise, right? So then they're obviously more and more expensive also, right? So thing is, there is an interesting cycle going on here. So you develop the next fancy experimental machine, right? Make more fancy observations, and advertise those observations in media. Oh, we were able to look into the depths of the cell and we were able to see the movements of a molecule, etc., etc. And lay people will think, oh man, how great it is. Right? And then, so these media buzz will be quoted, right, to when they apply for some funding. So look, if you have 1 photon microscope, you can observe so much. And if you imagine what you can observe with a 2 photon microscope, therefore give us more funding, we'll build a 2 photon microscope, and so on and so forth. So this cycle goes on and on. Point is, okay, you have made so many observations, you already have mountains of data. What have you learned from that? right? So I've seen a TED talk where this guy says, "I've observed 1,000 brains, and we have so much data now." Right? I mean, let us celebrate. But what have you learned from your observations? Right? If you haven't learned anything from 1,000 brains, what's the point of observing the 1,001st brain? So this goes on and on. Why is it like that? Why have observations become so important? I mean, right? So the thing is that something to do with the nature of biology itself. So neuroscience is naturally a branch of biology, right? And biology is basically, right, in its the way it has started is very descriptive, because the biological systems are apparently quite complex, and they look very complex with lots of richness and variety of form and all that. So when you observe them, it needs a lot of observational effort to describe them completely. So this picture, for example, is a very beautiful drawing by Antoine von Leeuwenhoek. He used this early microscope to look at some simple samples like an onion skin and some dirty water and so on and so forth. So you saw some cells in it. You saw some kind of little cells moving around, lashing their tails and so on. It was very interesting. How can such small objects move around as if they're alive, right? So he called them animalcules. It's like an animal plus molecule. So he made all these pretty pictures. So that's a hallmark of biological tradition. We literally look at things and draw pictures, right? Anton von Leeuwenhoek has drawn pictures with a pencil on a paper. Now we draw pictures with a fancy MRI scanner or a fancy microscope, right? Or manipulate small objects with atomic tweezers and so on. But so the tradition is still there, quite alive and quite kicking. So it is the same tradition which helped us in generating huge amounts of data about biological systems, right? Is also sometimes becomes it becomes a stumbling block. It becomes an obstacle. This point was very well described by Yuri Lazarnik in the article "Can a biologist fix a radio?" or "What I learned while studying apoptosis." So it gives a very interesting story, right? And somebody wanted to understand how a radio works, and they didn't know anything about the principles of radio, electrical engineering, nothing. So in a typical biologist, so this is a biologist, right? So in a typical biology approach, what this person has done is, this is obviously a fictitious story, which is a made-up story, just to see how a biologist will approach study of a complex system. So this guy, so he has ordered a lot of printed circuit boards of different kinds of radios, huge number of them, right? And then started analyzing them. And how would you analyze a biological system? You analyze a printed circuit board in the same way. So if you look at this picture, right, how would you analyze it? How would you describe it? Okay, I can see some blue cylinders with a silvery head, silvery top surface. See a couple of them. Then there's also a kind of a blue cube-like structure, and then kind of brown cubical structures. Then there are kind of bronze or brown and strange flattened cylindrical structures, and some cylinders have colored stripes on them, and some are prolate ellipsoids, and some are oblate ellipsoids, etc., etc. So I can make a very detailed description, measure their heights and lengths and diameters, and draw lots of pictures and so on. So this guy started doing that for a long time and for many years for different kinds of PCBs and so on. Okay, so that's a huge stage of research or a phase of research which is called structural analysis. I mean, that's what you do in biology, right? So the first thing you do is structural analysis. You study anatomy, you study the structure of molecules, and so on and so forth. A huge amount of effort goes into that. Then comes the second phase where you get into functional study, functional analysis. So how do you do that? So you want to look at the function of the printed circuit board. So you power up the PCB, you connect the circuit to the power lines, right? And then see what happens. See what does it do? Normally it will produce some sound, I guess. If you pick up some station, it will produce some sound. Now you want to understand which component on this PCB contributes to sound, in what way. So how do you do that in a typical biological fashion? You start pulling out the components one by one, randomly, right? And then note down, okay, if I pull out this component, what is the effect on the sound produced? Nothing. Okay, put it back. Next, pull the next one, and so on. Kind of like a monkey picking lice, right, from a I don't know, family member. So you kind of pull out these components one by one and see what is the effect of the knockout. This is called knockout genes in genetics on the output. So you do that, and after long last, and after years of such an effort, very painstaking effort, this guy finds that if you pull out some important wire, right, which probably carries power to the PCB, uh, there is no sound. Sound completely stops, right, because there's no power, and that becomes a major breakthrough. And it comes in all newspapers, and so this guy gets a huge funding for further research and gets faculty position offers from top universities, and so on and so forth. This is how the drama goes on. This is story. It's quite hilarious. You read that article. Point is, this is how if you approach understanding or study of a radio circuit in the manner of a biologist, the way biologists approach research. This is how it looks, and it looks dreadfully complicated. But whereas, how would you study a PCB? How would you study a radio? What does it even mean to understand a radio, the architecture of a radio? So first of all, we need to understand that radio is founded on the principle of detecting or picking up electromagnetic signals. So you need to first understand that there is such a thing called electromagnetic signal, all right, and then that will be transmitted by radio station by adding a message signal to a carrier wave, right? So the message signal rides over a carrier wave, and that's what carries the information from the radio station to all the receivers. Now, this signal is picked up by a circuit called the tuner circuit, which responds to the signal at a certain given frequency to which it is tuned. Then that is, so you demodulate that and get the signal out, and you amplify that signal, charge it up, remove some noise, clean up the signal, and feed it into a speaker, and out comes a song or a speech or whatever is being transmitted on the radio, over the radio. So this is the outline. This is architecture, essential architecture of a radio. If you understand that, you are mostly done. Then the question is, okay, what is the amplifier? What kind of amplifier? What is the circuit diagram of the amplifier? Right? How many dB? What is the gain of the amplifier? And etc. All these things you can study. So without understanding that these are the essential elements of any radio, if you just randomly launch into a kind of a valiant brute force study of radio circuit, right? It will take forever and never even understand how radio works. So we need this kind of a first principle approach. And I try to, because I come from electrical engineering and I try to understand neuroscience from the point of view of an engineer, and I try to teach myself in a certain fashion. So with that effort, I've written a book. So if you want, you can take a look at this book later. So this is the problem. We are living in this big data. So therefore, a lot of data is generated. And if you look at the pillars of science, of modern science, in the earliest period, actually, science began with experimentation, right? Theory was a lot of other things were there for a long time. A lot of them were very flaky theories. But mathematical theory is somewhat more recent. Empirical formulae were there for millennia. But more solid mathematical theory, calculus, all that is much more recent. So we began modern science theory and experiment. More recently, the last half century, we had computation added. A fourth pillar that people are talking about as one of the pillars of modern science is the data pillar. So that's become a big thing these days. So in neuroscience also, like I was just saying, data is a very big thing. And so there's a huge amount of data at various levels of abstraction, various scales in the brain. For example, since brain is a network of neurons, it's very interesting, fascinating cells called neurons. There are a lot of data about different kinds of neurons and all that. And neurons, like a typical cell looks like a fluid-filled blob, whereas a neuron, right, in addition to the blob, which is at the center of the cell, it has a huge number of wires sticking out. And there's arbors, right? You can call them dendrites, whatever. I'll talk about all that later. But you see a cell body, that little black dot in the center. There's a cell body, and around that you have a huge amount of wires sticking out. And the wiring patterns are different for different neurons, and some neurons have like very long wires, right? And some neurons are like thick shrubs. right? And some neurons have very thin wires and a small number sparse wires. So all sorts of neurons are there and with rich variety. So people have uh, measured all these mensurational properties of neurons, and then their wire lengths and branching patterns and the thicknesses of wires, their conductances, and name it. They have done all. They've taken all those measurements. So huge atlases are available, right? So this is one atlas like that called Neuromorph. It has lots, thousands of neurons of different kinds from different species and different parts of the nervous system, and all the data is available. What do you do with that? That's a different matter, but data is available. Similarly, there is a project called the Connectome project. It's a pretty ambitious project. It aims to work out the complete wiring diagram of the brain. That means all these 100 billion neurons of the brain, right? You want to, so they form a graph, right? Because these neurons are connected to each other by some kind of wire. So that forms a graph. So now what is that graph, right? Among these 100 billion neurons, which neuron is connected to which other neuron? That's the ultimate objective of the Connectome project, which is very ambitious. But people are not able to work out the graph at that level of detail, and I'm not even sure if it is necessary. That's a different matter. But people are able to come up with a more gross connectivity maps of the brain, right? There's lots of technology that's available right now to do that. And so instead of working out the connectivity of all the 100 billion neurons as individual cells, people are able to group those neurons into clusters and come up with the work out the connectome. But they've done it for smaller organisms, like for example the C. elegans, or also called the nematode. It's a very small worm which has about 300 neurons and 10,000 connections. So this nervous system has been completely worked out. Okay, so when it comes to humans, people are able to work out the connectome at a slightly gross scale. Instead of 100 billion units, you divide that into a few thousand units and then work out the connectome. So that kind of data is available. So the thing is, I mean, there's a huge race to work out the connectome because each trying to beat the other in terms of resolution and level of detail, accuracy, and so on and so forth. But the point is, what is the hurry to find the connectome? Because I strongly believe that conceptual progress should come first. Data can wait. I mean, we have whatever data we have we extracted the kind of the conceptual milk, right, out of that data first before we go on and get even more detailed data. So for example, we don't even know why there are two cerebral hemispheres. So why does the brain look like what it is, right? What is the logic? What is the meaning of its cross architecture? Instead of just working out and counting everything and measuring everything on and on and on, let's first ask why is it like that? Why is there the sheet of neurons called the cortex on the surface of the brain, right? And then other structures like cerebellum, for example. Cerebellum sticks outside the brain, right? Whereas other structures like thalamus are deep inside the brain. Why is it like that? What is the logic? What does it get by that kind of an arrangement? So these kinds of questions, asking why at every step and trying to extract deep theories of the brain, that process is somewhat weak right now in neuroscience, right? Therefore, the field is lots of data, but tiny theory. Why is it like that? Like I said, if you go back to the history of science, astronomy, we can draw some parallels. So there was a time when, so if you look at the night sky, let me tell you this small side story. So if you look at, if you just sit out at night and watch the stars, you'll see all the stars going in a nice circle, right? And the central circle is the pole star, right? So we believe that the stars are, the cosmos is basically a dark globe in which all the stars are studded, and this cosmic globe is constantly rotating while the Earth is stationary. So these people thought for a long time, and now we understand that the globe is not rotating. There's the Earth that is rotating, and that's why we see the stars rotating. So that's a different matter. So people thought that like stars move as if mounted on a celestial sphere, and the sun, moon, and planets move against the backdrop of that sphere. So so the sun and moon move with respect to us is quite simple, a simple circular kind of thing. But the motion of planets turned out to be a bit of a challenge, because planets, if you watch them every night, at certain times, say from the Earth, they seem to have very strange motion. For example, look at Mars. The motion of Mars, right? This is from 2003. it seems to go forward in the sky for some time and then go back. So here the background of fixed stars is like is what is used as a reference frame. So against that backdrop, Mars seems to move forward for some time and go back a little, and then again go forward for some time. So this kind of a backward motion is called retrograde motion. This is something very peculiar about planets. When you watch them from the Earth, if you look at sun or moon, that doesn't happen, right? What is so strange about planets? So you see that even with other planets, Jupiter, Mercury, Venus, and so on. Now we have lots of data, so you can see all this. So planetary motions, when you observe them from the Earth, look very wacko, right? And people knew it for a long time because people had observation astronomy for a long time. So how do you explain this? What is the world model which can explain this? So this guy called Ptolemy, Claudius Ptolemy, way back 140 AD, right? He thought that Earth is center of the universe. There's also a strong, some level of religious influence on that perhaps, but that influence not only remained but also became much stronger over the next 1.5 millennia, because much later this guy called Tycho Brahe was also an observational astronomer. Right, he also came up with a kind of a. So this is a purely geocentric model where the Earth is thought to be center of the world, and all the other heavenly bodies are moving around it. The thing is, in order to account for all this vacuum motion of the planets, this guy had to assume that the planets not only move around the Earth, but there's also a small internal motion, like a smaller circle. There's a big circle that they trace going around the Earth, and there's a smaller circle, right, which is superimposed on the bigger circle. So he called these cycles the epicycles. Because observations are so complicated, right, they had to come up with very complicated, a very complicated conceptual framework. And therefore, the mathematics also was abnormally complicated to account for all that. So he had to use very complicated trigonometric series and so on to capture this. This kind of a story that Earth is the center of the universe and then all the planets go around it, and then they have all this inner motion, the cyclic motion superimposed. This story stuck for 1.5 millennia, 1,500 years. And only thing is people would tweak their formulae, add one more higher order term just to make the observations more accurate. But there's no fundamental shift in the paradigm. That's a problem. What kind of paradigm shift came in astronomy with the theories of Copernicus? Okay, people say that Copernicus has got his ideas from all the translations that they got from ancient Greeks and who in turn got from Indians and all that, but we don't know about that. But this is what we have about Copernican model. So he said, no, Sun is the center of the universe, center of whatever, known universe for them. And Earth goes around it. Not only Earth, other planets also go around it. And therefore, if you accept this model, now if you imagine how a planet would look if you observe it from the Earth, you'll see all sorts of strange behaviors. You'll see it going forward for some time, backward for some time. And if you look at the planet from the Earth, with the stars, playing the role of a fixed background, right? Then you will see all the strange behaviors that you are seeing in planetary motion. So on the whole, the model became much simpler. The complex could be accounted for using a much simpler model. And on that basis, Kepler has kind of distilled all the planetary motions into some patterns, into basic patterns, the data into some basic patterns. And he formulated these 3 laws, which you might have studied in your high school. And then our man Newton has taken these laws and said in all these 3 laws, underlying them, there's a very simple mathematical principle. So there is some kind of a centralized force acting on the planets with the sun as the center. He called it gravity, and this force acts like follows some inverse square law. Okay, and then there's laws of mechanics which can be described in terms of calculus with differential equations. That's it. After that, you can describe the motion of all planets. In fact, that mechanical framework was found to be so successful for hundreds of years until Einstein came to the scene. And the same framework was used to discover new planets, to detect the possible existence of new planets, right down the visible ones, like Uranus, Neptune. They were all found based on perturbation because the observations and the material were not matching. So theory has to be correct, observations cannot be correct. And that was the level of confidence in Newton's equations. So the thing is, once paradigm has changed, the the theory becomes solid, right? And the observations can be accounted for. Until then, the observations can be accounted for by only very complicated formulations. So it's only when the paradigm is wrong, the descriptions look very complicated. So you see that there is a huge transition that was made from Ptolemy to Tycho Brahe. Ptolemy is 2000 years ago, Tycho Brahe is about 500 years ago, to Newton about 300 years ago. Right, the huge transition was made, and then that's why it is called, what Newton has achieved is called Newtonian revolution. Right, Newton has put all these ideas and really he has rewritten entire physics almost single-handedly, right, and has written and put all that into a book called Principia Mathematica. Right, he showed how powerful mathematics can be in describing the laws of nature, phenomena of nature. Now, I think there is a very interesting book, almost the Bible of neuroscience, called The Principles of Neural Science. So it'll be interesting. I keep wondering, if somebody can rewrite these principles of neural science in terms of Principia Mathematica, like Principia Mathematica. So where you have the basic laws of the brain in the very first few chapters, and after that you work out, apply these laws to various systems in the brain and explain their function quite thoroughly in great detail. The way you study like in any modern science, right? Not just empirical. So anyway, we haven't got there. We are not even anywhere close. But I want you to understand that there is a problem. So there's a problem, and there's a crisis in neuroscience. People might gloss over it, but there is a problem. We do have theories, like I was saying earlier, but a lot of them are like Ptolemy's epicycles. Their formulae, they fit the observations very well, but they don't look elegant. They are not universal. One of the beauties of the Newtonian revolution, he called his law the universal law of gravitation. I mean, what guts that man has, right? Ptolemy or Tycho Brahe never said their formula are universal. This is a formula for Jupiter. This is a formula for Saturn. This is a formula for Mercury. This is a correction for the formula for Mercury, and so on and so forth. Now, there's nothing universal about them. That once Newton has found his equations, he said they are universal. So that means not only for our solar system, for any solar system anywhere in the universe, they should work. That was his confidence, right? And we don't have that kind of confidence with most of the neuroscience theory that is present right now. It will work for any brain, for any species, any future species. Nothing like that. We are not even close to that kind of a confidence. Okay, so we have a lot of co-fitting data, co-fitting models, but the kind of fundamental laws of the brain, we're still struggling to find out. There's some hints at it, but we really don't have it yet. So neuroscience today is theory weak. That is no comprehensive, satisfactory theory of most brain structures, and a lot of hand-wavy theories. And those explanations are actually non-explanations. They sound like explanation, but actually non-explanations. So some of the neuroscience people in the community are also feeling the pinch of it, feeling this kind of a deficiency, this vacuum, and commenting on it. But the mainstream is too happy with what's happening. So they go on merrily in a collecting more data, writing more papers, and so on. So V.S. Ramachandran, Indian origin neuroscientist who is based in University of California, San Diego, he says that we are at Faraday's stage in neuroscience, where you have lots of empirical observations and some kind of working operating laws. They're not really deep laws. And Faraday came before Maxwell, so before Maxwell equations. So we are not at the stage of Maxwell equations. We are at the stage of Faraday and Ampere and all these guys. So Patricia Churchland, the neuroscientist and the neurophilosopher, she also says that there are no neural theories, no good ones anyway. So, so that is the thing. And again, let me ask, why is it like this? So I gave some reasons because of the descriptive nature of biology and all that. Another reason, which is also very common and which is becoming more and more entrenched in modern biology, is the problem of reductionism. So in any piece of matter, right, or in any biological system, you'll have multiple levels of abstraction. You can describe it at multiple scales. At the smallest scale, anything will have just molecules or atoms if you go even smaller. Then in a biological system, if you go to slightly higher level, you'll have cells. In case of brain, it is neurons. Then if you go to even higher level, you'll have systems. That is the part of the visual system or somatosensory system or olfactory system, motor system, and things like that. So there are all these levels of description. So reductionism is a kind of approach or a philosophy or a belief system that says that you can only explain a function of a system only by reducing it to its smallest constituents. You have to describe it in terms of the smallest constituents. Only that is a valid and acceptable description. This is the position of reductionism. But that won't work. It's unnecessary kind of, it makes the problem unnecessarily complicated. Right? In engineering, for example, we don't use that philosophy. If we use that philosophy, we'll never have any engineering. Take, for example, the problem of designing or studying an aircraft wing. You want to study the effect of the wind on the aircraft wing. So what do? You write the Navier-Stokes equation, right? And to interpret the data. What is it? So what does the wind constitute? Right, constitutes of essentially molecules, the air molecules. But point is, we're not going to describe the air molecules, the collisions of the air molecules on the wing and all that, because that level of detail simulation will take probably tens of years. We don't do that. We don't go for that foolishness. We invoke this abstraction called fluid. Right? Fluid is an abstract thing. What is fluid? I cannot. It's not like a particle. It's something more than a particle. It's a mass of particles which has its own properties. It has properties like density and volume and uh, uh, viscosity and things like that, right? So you can describe it very rigorously mathematically, but it's slightly abstract and it's not molecules. And using this, you can finish your design of the wing in a reasonable amount of time. So we are not following a reductionist approach here. We are describing the phenomena at a middle level. We are not going all the way to atoms and molecule level, right? Because that will be nightmarish if we take that approach. But in biology, we do that all the time. We want to be able to explain everything in terms of cells, which is kind of like the smallest constituent in a biological tissue, right? Or even better, molecules, because in the current biology, molecular biology is the queen of sciences, right? And everything else has to be ideally explained in terms of molecular biology, biological phenomena. So therefore, in modern neuroscience, you have tons of data at the level of single molecules, protein function, at the level of single neurons, single cells, and so on. But when it comes to large-scale systems, how does the hippocampus work? How does cerebellum work? Right? There's a lot of confusion, and you'll find typically lots of schools or camps, or schools of thought, right, represented by different labs in the world. Each lab keeps on describing a given brain system in their own way, and other labs describe it in their own unique way, and there's no consensus. So this goes on, and the confusion goes on. Confusion lives for a long time. And because of this problem, if you look at any of the major disorders, major brain disorders, we don't have a good theory of them. Why does it happen? What is it? What exactly happens in the brain? You might have mountains of data, all sorts of data, but we are not able to piece all the data into a coherent model, into a coherent story. And therefore, most brain disorders, bipolar disorders, schizophrenia, Parkinson's, Alzheimer's, ALS, epilepsy, I mean, name it, right? There's not a single of these major disorders where you can say, I can cure it. You cannot cure it, you can only manage it, you can give palliatives, you can suppress symptoms a little bit, but the problem remains, right? Which is very unfortunate. So it's like the elephant in the room. Everybody knows that it's a huge problem in neuroscience, but you just turn away from that and talk about some detail. What is the response of this cell to some stimulus? Who cares, right? So because of this, what is happening is, in contemporary neuroscience, basic neuroscience has huge volumes of data, but we don't know what to do with it. We don't even know what is the light it is shedding on the problems that we are interested in, which is the social problems, human problems that humans face, that is clinical neuroscience, or neurology, neuropsychiatry, and all these areas. That looks like almost an empty vessel. So I feel that if this problem has to be solved, we need to change the approach, right, to studying brain function. We need simplified models. So that's how it worked in physics and engineering. I think why not apply a similar approach even to biology and neuroscience? That's the way, right? And we need to develop the right kind of abstraction. So we've been working on these lines, for about 15 years, 20 years, whatever. Initially, we didn't have any idea. We were just blindly groping and stumbling and trying out many things. But over a period of time, we were able to evolve certain approach to modeling brain function. We call it, it's a triple formula. We call it simplify, unify, and apply. Simplify means when you develop a model, right, you have to simplify it to the point that any further simplification makes the model absurd. It is totally meaningless. So you have to take it to that most minimal point. So take, for example, this picture of Mahatma Gandhi. That left picture is quite detailed. You can see him, you can see his features and glasses and everything. But the right picture also, anybody in India, if you see that right picture, it's like a cartoon picture. You can immediately kind of connect that, it's recognized that it's Gandhi's cartoon. So it's a minimal model. So can we come up with that kind of minimal models of brain systems? So that is the first step, simplify. Then unify. What does unify mean? If the model is to be accepted as a valid and useful model, you should be able to explain a wide variety of experimental data, right? So you look at lots of different birds, and what is the birdness of all these birds? So that is represented by the simple cartoon picture in the center, that pretty bird. So that is unified. The third step is approach. So once you have such a unified model, a simple model, it will be very powerful. It will have a huge amount of explanatory power. So with which now it will be applied to many different problems. And what is interesting is nice, powerful neuroscience models can have applications both in clinical neuroscience, right, in developing better treatments for diseases, neurological diseases, and they can also be applied in engineering for developing better engineering systems. They can enhance the power of the modern AI and deep learning and all that. There's a lot of hope in the, recently that we understand brain better and use that knowledge to develop better models and better AI systems. And it can be even more powerful. It will be free from some of the problems we are facing with AI systems as they are today. Okay, so with that kind of approach, we want to model the brain, whole brain. We call this the Mesobrain. The idea of a Mesobrain or this mesoscopic level. Several neuroscientists have used this term. One of them, most prominently, is a man called Walter Freeman, who is no more, but I really admire this gentleman. So he used the term mesobrain quite a bit. So this level of this slightly simplified model of the whole brain, where you don't model it in terms of 100 billion neurons, but only maybe 10 million units, right? Where each unit represents lots of neurons. So with that, at that slightly abstract level, we would like to model the entire sensory hierarchy, entire motor hierarchy, and these two meet at this big decision-making areas called the prefrontal areas in the brain, and then from there are three loops: the cerebellum, basal ganglia, and hippocampus. Okay, obviously I'll be going into greater depth about all these things later. Right. So I feel that neuroscience, if you take this kind of integrative approach, that you'll get much better outcomes. So computation modeling or theory building must be the center, must occupy the center stage. Uh, and then other sources of experimental data, like behavioral data, neuroimaging data, electrophysiology. All this should feed into that computation modeling. So that means a single model should be able to talk to data from different sources, right? And then you should develop a model which is simple and unitary and so on, the way I've described just a moment ago. And using such models, you need to develop disease models to explain different diseases, and then develop treatments using such models. And that can be a basis for creating more startups. Using such startup, you should develop clinical applications. So this is the kind of vision we have, right? So coming to the course outline, just want to say that tomorrow will be history class, but after that I'll cover some mathematical preliminaries. I'll cover some basics of dynamical systems and nonlinear oscillators. After that, I'll start single neuron basics. I'll start with biology aspects. That is, what are the parts of a neuron, an axon, dendrite, and all that. Then what is the membrane potential, action potential, neurotransmission, all that stuff. So then a big part of the course is modeling the neuron at single cell level, the single neuron level. So here again, I start with Hodgson-Huxley model and then proceed to what is called the biophysical neuron model. That is modeling a single neuron, all its branches and all that in great detail. Then I say that, okay, it's too complicated, let us simplify it further, and then I present some of these two variable models of neurons, like for example, Fujii-Nagumo model, Morris-Likar model, and so on. Then I go to networks. I start with simple perceptron and multi-layer perceptron deep networks. There's a lot of work of late where people have found that deep networks can be used to explain a lot of sensory function in real brains. So I'll progress with that. Then Hopfield network, which is a recurrent network, and how it can be used to explain memory and hippocampal function. Then competitive learning and self-organizing maps. How do you explain brain maps? right? I just want to, then I'll talk about modeling brain systems. So single cell networks and then systems. That's how it will progress. So I'll talk about baseline hippocampus and Parkinson disease, hippocampus and spatial navigation, stroke and rehabilitation, and universal coupling. These are the main interests of my lab. So I'll talk about some of the systems level models. Grading midterm 30, assignments 30, and some 40. Some differences. So I'm using different books for different parts of the course. For the biophysics physical modeling part, I'll be using Christoph Koch's book, Biophysics of Computation. And all the notes is available on the net, my notes. So that's is mostly enough. But if you want, you can also refer to these books. Then for dynamical systems part, I'm using Izikevich textbook, a really brilliant book. You should read this. Then for new network part, for some of the topics, I'll be using Satish Kumar. There's also not that many great textbooks of computational neuroscience. There's one very nice book by Thomas Rappenberg called Fundamentals of Computational Neuroscience. So I myself don't use it, but I've seen the book. It's a very nice book. If you want, you can take a look at it. Right, and that's all for today. If you have any questions, I can answer it. Any questions? Guys, are you there? Awake?
Yes, sir.
Yeah. So the classes will be on Monday, Tuesday, and Friday. The Wednesday 8 o'clock class I'm not taking.
So what are the timings?
That's what I said. Monday to that's a C slot timings. Only Friday we need to decide. Apparently there is some confusion. Friday, Sandeep, is it 12 to 1 or 1 to 2?
Yeah, so 12 to 1 or 1 to 2, that we can decide. Maybe we will send a poll, and then
Yeah.
whichever majority prefers, then we will have that time. Anyway, it will be lunch break in 1 of the hours.
Yeah. So let me know. I'm okay with it. Any other questions on the content of the class instead of all this logistics stuff?
Sir, would we actually be doing any programming projects, or like would the assignments be theoretical, or would it also involve actually writing code and working
Yeah.
on actual systems?
Yeah, they will be programming assignments only. Mostly people use Python, right?
Yes, sir.
Yeah, they count for 30 marks.
Okay. Thank you, sir.
Yeah, because just by theory you cannot do much. You have to code. You have to write programs and see what happens.
So how many total assignments will we have?
2 or 3, right, Sandeep? That's what you have done last year.
Yeah, three assignments.
3, yeah. So that's it, right?
Yes, sir, that's it.
Okay, so see you tomorrow. See you all tomorrow. Have a good day.
Thank you, sir.
Thank you.
Thank you, sir.